Dear students, today practical session about the dental pulp histology. I'm Dr. Hibal Zar. I will be your host for this lecture. As usual, we will start with the lecture outline. We are going to discuss the organization and composition of the dental pulp that is actually composed of 75% of water and 25% of organic material, that is cells and extracellular matrix. These are the building blocks of this tissue. In the second part, we will discuss the blood vessels and the nerve supply of the dental pulp. In the third part, we are going to discuss different regions of dental pulp. And finally, we will discuss age changes related in the dental pulp. So let's go. When we take a section of the dental pulp to look at under the microscope, we will notice that on the outer surface of the dental pulp, there's a layer of odontoblasts sending their processes inside the dentinal tubules, as explained in the last lecture and in the last week. Underneath is a loose connective tissue that is filled with cells, blood supply and nerve supply, and of course lymphatics. In histological sections, we will see this layer of odontoblasts with the loose connective tissue underneath. We will discuss both of them in details. So, in the crown, if we take a section from the crown, you will see that the pulp is in connection with the dentine, with this loose connective tissue underneath. Inside the pulp, we will have this loose connective tissue with the blood supply and different kinds of cells. And as you can see, we can see fibroblasts, stem cells, nerve fibers. These all will be discussed in details. And in the root, remember always that after the dentine layer, there's cementum that's going to be discussed in details next week. What kind of structures can we find inside the dental pulp? So we will see different cells. First of all, the odontoblasts, and the main kind of cell we are going to see is the fibroblasts. They are the most ubiquitous cells in the dental pulp. We will see some kinds of course, and the stem cells that forms or regenerate fibroblasts and odontoblasts. They have a lot of different populations that I'm not going to be discussing at this level. A lot of immune cells like the T cells, the macrophages, the dendritic cells, antigen presenting cells, all of these are going to be seen in histological sections. We have some neutrophils, but they degrade under the environment we prepare histological sections with, so it's really hard to see them. And we are going to see, of course, the nervitic supply, the blood supply, and the lymphatic vessels. These are the components of the dental pulp that we are going to see and discuss today. The odontoblasts, as you can see here, they are polarized cells, which means that the plasma membrane make different compartments inside the cell, different compartments that have different functions. They are related or adhere to each other and to also fibroblasts and the dental pulp by different junctions that were discussed thoroughly in the lecture. Please go back to read about them. And of course, they have tubules extending into the dentinal tubules. As you can see here in this histological section, when we cut the tooth in an angle, these cells looks like they are overlying each other, giving this pseudo-stratified looking feature. They are actually not pseudo-stratified in the real histological section, or in the real in the real tooth, but they look pseudo-stratified in the histological sections as they are cut in an angle. You can see the dentinal tubules, of course, here, and the loose connective tissue underneath. That will be discussed in details in a few minutes. So the second kind of cells found in the dental pulp are the fibroblasts. The fibroblast usually has many shapes. A spindle shape, as we can see here, and the stellate shape, like a star sending processes, are the most seen shapes of fibroblasts in the dental pulp. So these cells are the most ubiquitous or found cells in the dental pulp. They have many functions. They make the extracellular matrix, the ground substance. They degrade it and turn it over. Also, as I have said, we're gonna see the macrophages, the dendritic cells, and the T lymphocytes. These are really hard to see and detect in the dental pulp. That's why there's only this one slide showing the dendritic cells here in close relationship with the odontoblasts, sending its dendrites inside the odontoblastic layer, and actually they extend into the periodontium. If there is a carious lesion, the dendrites can go actually into the circumpulpal dentine. So these are actually the components of the dental pulp, the cells and the extracellular matrix. The components of the extracellular matrix are discussed thoroughly in the theoretical lecture, and it's more related to cytology and biochemistry. That's why you have to go back to the book and read all the details about them. In relating to histology, that is all what you have to know in the first part, which is the components of the dental pulp, the cells and the extracellular matrix. And we will see you in the next part.